Hello, and welcome back to From Bench to Bedside. I'm your host, Dr. Melanie Matheu. I have a PhD in the field of immunology, and I love to take people through the history of diseases, how they were discovered, advances through time, and where we are today, and maybe what's coming up next. So that's what we're going to do today, and we're going to talk about CRISPR gene editing, sickle cell disease, and the two drugs that were approved this week by the US FDA. And this is some pretty interesting stuff. It's groundbreaking technology meets underserved disease. I think this is going to be a great story. Why is this super exciting to me? Why did I want to jump on this? Because I'm not jumping on every drug approval from the FDA. It is a first, so that is a big deal. But there's so much that has gone into this throughout the years. There have been no new ways or really good ways to treat sickle cell disease in the past. So this is a major, major advancement. This treatment is potentially curative, and that is very exciting. When I look at the broad field of health and medicine and all the things we could do, when you get to something that's curative, that is the most exciting. Something where people can resume their normal lives, go about their day, and not have to be feeling like they're tied to a pill bottle or tied to an injection. Curative is really, really exciting. I think we should all be striving for those curative treatments. This is the first approval for sickle cell disease that uses gene editing technology. And it's been a long time in the works, but also moving pretty fast. So we're going to talk about that. What's not exciting is the price tag for this. And I'm going to talk about that towards the end. But let's start with what is sickle cell disease. And I apologize for those of you watching me on YouTube for the visuals. I have them there. If you guys listening on the podcast want to follow along with some visuals, I'm going to be scanning some of my notes. So this is a big, big topic. And I wanted to make sure that everything was well fact-checked, well researched. You will find the references in the show notes. And let's get going. All right, so let's talk about some of the demographics of sickle cell disease. The number of people living with sickle cell disease increased 41% from 2000 to 2021. Across the world, it's something like an estimated 5.5 million in 2000, in the year 2000, to close to 8 million in 2021. So that 8 million is considered an underestimate. I've seen numbers as high as 20 million, but like a lot of underserved diseases, it's hard to get the accurate numbers from around the world. This is a large disease. We can look at millions and millions of people that are suffering from sickle cell disease. The median survival is about 45 to 55 years. It shaves about 20 years off of people's lives. And it is expected to continue to increase around the world. Some groups estimate by about 30% between 2010 and 2050. The number of total people living with sickle cell disease in the United States is unknown. Approximately 100,000 are estimated, and it's about 1 in 365 Black American births. They have sickle cell disease, those babies, okay? And in Hispanic Americans, it's about 1 in 16,000, so the rate's a little lower there, and about 1 in 13 Black American babies have sickle cell trait, and we'll talk about that. So that's at a higher rate, but not associated with disease. But it does mean that those children will be carriers of that disease. And if they marry someone else who has sickle cell disease or sickle cell trait, it is likely possible, not guaranteed, but very likely that one of their children may have sickle cell disease. Sickle cell disease is known as being autosomal recessive. That means you need one copy of a gene from each parent that had either sickle cell disease themselves or sickle cell trait. And so if one parent has sickle cell trait and the other parent has sickle cell trait, there's a 25% chance that their child will have sickle cell disease. If both parents have sickle cell disease, it's a 100% chance that their children will have sickle cell disease. For those of you following along on YouTube, I've put up it so you can see what I'm talking about with that genetic distribution in case you didn't take genetics since high school. Sickle cell trait and sickle cell disease are more common in certain groups. So this includes African Americans, Hispanics, South Asians, Caucasians, people from Middle Eastern countries, people near and around the equator where malaria is prevalent. So why is that? Sickle cell trait, that means one copy of the gene that causes sickle cell disease but does not cause severe disease itself, can provide protection from malaria. Having sickle cell disease, on the other hand, has higher mortality rates associated with it and a lower chance of surviving malaria. So one copy of the gene is protective. And people who have sickle cell trait, during infection, they have 50 to 90% fewer parasites in their blood than people who have normal hemoglobin. And the protective effects are seen strongly when they're young children. Why is it called sickle cell disease, and when was it first discovered? So in 1904, a medical intern named Ernest Irons was examining the blood of a man named Walter Clement Noel. He had developed respiratory problems. He had ulcers before. This is something else that happens with sickle cell anemia and sickle cell disease. And he was this 20-year-old dental student for Grenada. He was born in the West Indies. And this is kind of a new medical procedure at the time, but they added a blood smear to his workup. They looked at his red blood cells under the microscope. This was 1904. And Irons kind of described on the blood smear that there were many pear-shaped and elongated forms of the red blood cells, and some of them were very small. And this was odd. This did not look like other blood. In 1910, Dr. James Henrich at the Chicago College of Dental Surgery published a formal paper describing the differences in shape of the red blood cells from Walter Clement Noel, but they really did not know what was going on. They really didn't have an idea of what could cause this, but they had a hunch it was what was ailing Mr. Noel, the student. Later in 1927, Hahn and Gillespie associated the sickling, the formation of a kind of elongated red blood cell shape with low oxygen concentrations. And in 1940, Sherman reported that the sickling of these red blood cells in the absence of oxygen was caused by a change in the molecular structure of hemoglobin. Hemoglobin is the main oxygen carrier. That is, the red blood cells are filled with it, and oxygen goes in and binds to it in high oxygen environments like the surface of your lungs. And then when you go to low oxygen environments out in your periphery and the capillaries and the microvasculature of your system, it gives up the oxygen because there's a lower oxygen concentration out there. So that's how that works. But in those low oxygen concentrations in the capillaries, those cells were changing their shape in a way that wasn't helpful to anyone. So in 1949, the chemist Linus Pauling, very famous for other work also, published in Science Sickle Cell Anemia, a molecular disease. He was the first to call it a molecular disease. He was able to show that the hemoglobin was indeed different from normal hemoglobin. So what's going on? Why was there some change, right? At this point, scientists understood that low oxygen led to a change in the shape of the red blood cells, and that this had to do with the hemoglobin molecule itself. Something was different. Something was going on there. So what is going on? So there's a small mutation in the hemoglobin that when it changes its shape, when it gives up its oxygen, it has a shape change, and that exposes a hydrophobic patch. So hydrophobicity means it doesn't blend well with water. It's not too soluble anymore. And if you have two copies of this gene, enough hydrophobic patches are exposed that they start to stick together, almost like magnets inside the cell, and they line up all the hemoglobin molecules and they create fiber. And this pushes the red blood cell into this elongated shape that is stiff and it's sticky and it clogs up capillaries in ways that aren't great. So remember, it's going to give up the oxygen when you're out in the peripheral system away from the lungs. And it's going to cause all sorts of little clotting and damage to a lot of tissues that rely on those capillaries for nutrient and oxygen exchange. And this is what leads to severe physical pain, fatigue, shortness of breath, dizziness, and a lot of damage to organs, damage to kidneys, damage to the lungs, and changes even in bone structure. So we think of bones as kind of a hard, static thing. They feel very tough, right? But they're living, they're supplied with oxygen, and you have all sorts of cell types, including stem cells, that live inside your bone marrow. And because the sickled red blood cells can damage capillary blood flow, you get different shapes in sometimes the vertebrae of the spinal cord. There's a classical H shape that's seen on the radiology. And then you can also have atrophy of the femur head. So people can have hip problems from sickle cell disease as well. But remember, one copy of this mutation is very protective. It's so protective that independent mutations that cause sickle cell disease arose in multiple places throughout time. In fact, 3 can be found from Africa and 1 was found in India. So this was so protective that it was selected for in the human population. Unfortunately, having 2 copies of this gene can be very detrimental to your long-term health and just your enjoyment of life. So that's high morbidity is what we would say. So you really struggle. You don't know if you're going to have an episode, when it's going to come on. Lots of things associated with sickle cell disease, even in young children. We're talking the onset of stroke as young as 6 years old, the necrosis of the bone marrow and the bones, pulmonary hypertension, pulmonary fibrosis, deteriorating renal function, severe anemia, acute chest pain. And then as kids kind of hit the adolescent period, this is when it can get really bad for them and they really need a lot of support. So that's when you start to see ulcerations in the legs, the mouth, increasing levels of bone pain crisis, delayed growth and delayed sexual development, and all sorts of other issues. I've even read about hair loss. It can be very, very detrimental. And then over the years, the cumulative organ damage, pulmonary fibrosis, hypertension, and progressive kidney disease. And that's usually what ends up cutting people's lives short. Also, people have, because of the pulmonary fibrosis, a lower ability to fight off a lot of bacterial infections. All right, back to 1956. In 1956, it was discovered that it is a single amino acid change, not even a whole stretch of the protein that is changed in the sequence of the hemoglobin gene, hemoglobin B, that causes sickle cell disease. In 1968, the Black Panther Party for Self-Defense, it was founded in 1966, but in 1968, they began to establish people's free medical clinics because of systemic discrimination against Black people in hospitals and private medical practices. And this is a really neat program. It offered everything from free breakfast kids for school kids and free sickle cell anemia or sickle cell trait testing. And by 1970, all chapters were required to establish a people's free medical clinic. I think that's truly incredible. In 1971, the Black Panther Party established the Sickle Cell Anemia Research Foundation with efforts and direction towards eradicating the disease. And I think that is just truly phenomenal. It's a testament to what people can do when they organize. In 1972, because of their lobbying, the U.S. Congress passed the National Sickle Cell Anemia Control Act. And this is really exciting because it created national genetic counseling, testing, and established research to diagnose and treat sickle cell disease. In 1975, the state of New York became the first state to add sickle cell anemia to its panel of newborn screening tests. Today, across the United States, sickle cell disease is tested for. It was called sickle cell anemia back then because it was just being thought of as one, but sickle cell disease really covers quite a few different incarnations of the genetic mutations. Now, across the United States, sickle cell disease is tested for in all infants. In 1984, a child who had leukemia was given a bone marrow transplant to save their lives, and it turned out that that ended up carrying their sickle cell disease as well. So that was a side effect of the treatment for leukemia, but it demonstrated that there may be a path to a cure. In 1995, a study was done using hydroxyurea in sickle cell, and it showed that there was definitely an improvement. Also in 1997, it was shown that periodic blood transfusions in children who have sickle cell disease, who are at risk for stroke, reduce the risk of stroke by 90%. So that was huge in 1997. Under the Orphan Drug Act of 1983, the U.S. Drug Administration approved hydroxyurea for sickle cell disease. And that was pretty exciting. That was one of the very first treatments around. In 2017, not too long ago, the U.S. Food and Drug Administration gave full and final approval for the use of hydroxyurea in children to treat sickle cell disease. Between 1998 and 2017, the drug had only been given special use permission to treat children. In 2017, the go-ahead was given to use medical-grade L-glutamine to treat children as young as 5 years old. In 2019, the FDA approved 2 more drugs, one from Global Blood Therapeutics for adults and children ages 12 and older, and then one from Novartis that was ideally targeted to reduce the number of vaso-occlusive crises. So that's an episode for someone who has sickle cell disease, and that is for adults and children aged 16 and older. Now, a lot of these therapeutics did represent significant advancements in the treatment of sickle cell disease, and one came from a very surprising place. So there was a reduction in the deaths of children, significant reduction with the development of a 7-valent pneumococcal vaccine in the year 2000. So I'm going to read you some relative death rates and their changes from 1983 to 1986. Versus 1999 through 2002. So ages 0 to 3, and these are people who have sickle cell disease. Ages 0 to 3, a 68% reduction in death. Ages 4 through 9, 39% reduction in death. And ages 10 through 14, a 24% reduction in death. So just by developing a vaccine, it was able to protect those children from getting severe lung disease that could lead to death. So I think that was really neat. So that was all going on. And then in 2019, the FDA approved 2 drugs to treat sickle cell disease, one from Global Blood Therapeutics and one from Novartis. The one from Novartis specifically targeting the vaso-occlusive crises. It was meant to reduce the number of those. But something else was going on in 2019. The very first hematopoietic stem cell transplant, autologous hematopoietic stem cell transplant. And autologous means it's from you, it's your own, with CRISPR edited hemoglobin to remove that one protein mutation, that one amino acid mutation was done. So what the transplant was is that stem cells were harvested. The patient underwent chemotherapy to remove their stem cells that were still there, kind of like you do for leukemia treatment. The gene was edited outside in a lab. The stem cells were grown up and then given back to that person. And she's still alive today, and she is not having the vaso-occlusive events, the episodes that she had before. She is quoted as saying this changed her life. So what the heck is CRISPR? I'm going to walk you through it. So CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. That is a mouthful. So CRISPR, you can see why scientists named it something else. But it's a gene editing technology that comes from the adaptive immune system of bacteria. Bacteria that co-evolved with viruses, just like we all have. There's something called a bacteriophage, and bacteria need a way, even though they're single-celled, to fight off the bacteriophage that try to invade them and replicate in them, just like they do in our cells. So their immune system is based on what we call CRISPR. So how that works is CRISPR is bound to something else called Cas. Classically, kind of Cas9 was the first one we really talked about a lot in research. And they're both expressed by the bacteria, and they identify and bind to target sites of the bacteriophage's genome as the virus tries to get its genome replicated in the bacteria, and creates a double-stranded DNA break, SNP. Okay. And there are lots of different CRISPR complexes now. We know it can attack RNA. We know it can do a lot of other things. But this is lethal to the bacteriophage. It can't replicate anymore because it has this CRISPR-Cas9 complex coming along and snipping the double-stranded DNA. Now I'm going to give you a brief history of how it was discovered, and this is brief. When I talk about one person, just know there's probably 50 or 100 scientists behind them working in labs, doing good research, publishing around the same time. But this research that started not too long ago and ended with a Nobel Prize in 2020. So a lot of the story I'll be pulling from the publication CRISPR-Cas9, a history of its discovery and ethical considerations for its use in gene editing. And you can go ahead and look that up. It's a really fantastic write-up published in 2022. All right. So way back in 1987, Yoshizumi Ishino, I think I'm saying that right. If I'm not, I'm going to apologize for every pronunciation because this is truly a global effort of scientists. But Yoshizumi Ishino, a Japanese molecular biologist at the University of Osaka in Japan, discovered the DNA sequence of CRISPR in bacteria. And he published on his findings in the Journal of Bacteriology, but no one really understood the significance yet. He kind of found this and he's like, this looks very odd in this strain of bacteria. I'm not sure what to make of it. I'm going to put this research out for the world. And this is what scientists do. We collaborate around the world when we see something new and interesting in our field to try to figure out what's really going on. And then we had Francisco Mojaca of the University of Alicante in Spain. Again, I apologize for my California accent in pronouncing all of your beautiful names. He found that there were similar structures in a totally unrelated strain of bacteria in 1995, just completely different evolutionary path, different bacteria, wildly not related. And so he was the first to say, hey, these might have some real functional significance. We're finding these repeats conserve, and we should look more deeply into this. Mojica was, in fact, the first scientist to hypothesize that these unusual loci that included fragments of foreign DNA that he found in archaea and were also in bacteria may be part of an ancient immune system. Then the first real experimental information about the CRISPR system came in 2007, and not from academia, from an unusual place. There were two French food scientists who worked with yogurt cultures and for the company, the Danish company Danisco. And due to the company's rich collection of bacterial strains they've been collecting since the 1980s, the scientists were able to trace the historical course of bacterial acquisition of the spacers at the CRISPR locus in response to viral attacks by bacteriophages. And so when that DNA is snipped, the bacteria end up incorporating some of it. And so they could trace that through the evolution of the bacteria and as they were warding off these bacteriophages. Another crucial player in the CRISPR-Cas9 system was a short RNA molecule, and this was discovered by John van der Oost of Wageningen. Oh boy, I'm saying that wrong. University in the Netherlands. And he was the first one to describe the existence of these RNA molecules in CRISPR-associated RNA. And then we have a researcher at the University of Lithuania, Vilnius, Lithuania, Dr. Sickle was the first to demonstrate the length of the actual guide RNA to be 20 base pairs. And this is how the CRISPR-Cas9 system knows where to make a snip, knows where to cut. And the final piece of all of this was another short RNA molecule discovered by Emmanuelle Charpentier, and for that she shared the Nobel Prize with Dr. Jennifer Doudna, who joined the race with multiple other labs Once all of the molecules and the known components of the CRISPR-Cas9 system were discovered, there was a massive race, a massive scientist race to get this working in mammalian cells because the power of this could be tremendous. And so this was an absolute dead race to get this done. And there were a lot of interesting publications that came out of it very quickly. So 5 research articles almost simultaneously in 2013 came out where each team reported that they had achieved the goal of getting CRISPR-Cas9 to work in human and animal cells. So we had the laboratories of George Church at Harvard, Feng Zhang, who was at the Broad Institute at the time. He is now at MIT. And they were showing that it was successful for DNA editing in human cells. And it was necessary to have several steps and codon optimization, etc. There was also Jennifer Doudna's group that showed it could be done in mammalian cells. Jin-Soo Kim at Seoul University in Korea, and Jennifer Doudna's at Berkeley here in California, and J. Keith Young at Harvard School of Medicine. So this is just a race to see who could do it best, who could do it fastest, who could do it first in human cells and most successfully. And there were amazing contributions across the board to make this happen. In 2020, Jennifer Doudna of the University of California, Berkeley, and Emmanuelle Charpentier, and she was a director at the Max Planck Institute for Infectious Biology, were awarded the Nobel Prize for their contributions and discoveries here. Other researchers have continued their work both on the CRISPR systems as well as other phenomenal research. George Church does a lot of really interesting science. I suggest you look him up as well. But improving upon this initial discovery has been a focus for many because the CRISPR-Cas9 system wasn't perfect. It was a start. And Feng Zhang, even as early as November of this year, 2023, his group at MIT published a paper finding 200 new functional systems that look a lot like CRISPR. And they can all be developed into, the best ones, of course, into better, perhaps next generation gene editing technologies. And I believe his group's paper is published in Science, November of this year. So that was a lot of scientific history, but I think it tells a really interesting story of how science often happens. Way back in 1987, we had a publication come from Japan where a scientist said, not eureka, not I found it, but that's interesting. And I'm loosely referencing writings from Gordon Taylor, a UK journalist, and Isaac Asimov, who said something similar, that the most exciting phrase in science isn't eureka, it is, that's interesting, that's different. But that started way back in 1987 and slowly built over time as pieces of the puzzle were added, and then finally touched off a Formula One style race to the finish where we had true gene editing technology advanced. Now, what do we do with it? And this is the point where that kind of amazing scientific technology collides with an underserved disease that doesn't have a cure or really any good treatments. I remember back to when I talked about that child that was cured from a hematopoietic stem cell transplant. That is known to be curative for sickle cell disease. The problem is it's really hard to get donor matched. And if you are of African descent or East Asian descent and you are in the United States, it is particularly difficult to find a bone marrow match because you are in a minority population. There aren't as many people to scan through to look for a bone marrow donation that will match. So why did a hematopoietic stem cell transplant cure that child of sickle cell disease? Well, hematopoietic stem cells sit upstream in the developmental process of creating red blood cells. All your red blood cells come from hematopoietic stem cells that live in your bone marrow. So if you can change that one amino acid mutation in your hematopoietic stem cells, you can change it for all the red blood cells in your body. And that is how this cure was approached. Can we take stem cells from a patient their own stem cells. So they won't be rejected. They don't need a bone marrow donor. We can do it for themselves. They can live without immunosuppression because they're not getting it from someone else. It might be a slight mismatch. They're getting it from themselves. We're going to change this one amino acid, this one tiny part of their protein, and give them back their own stem cells. And that is the basis of the therapies that Bluebird Bio and Vertex Pharmaceuticals developed. Both those therapies were approved this week by the FDA. So let's go through some of their data and what they found. These were not huge clinical trials. So I'm just going to say that it was around 30 to 45-ish patients in the different clinical trials. So for Vertex, their clinical endpoint was 12 consecutive months during a 24-month, 2-year follow-up period where they had no sickle cell events. None of the major sickling and disease was supposed to show up. And they achieved this for 93% of their patients. The effectiveness of the therapeutic for Bluebird Bio was evaluated on complete resolution, so no events, between 6 and 18 months after infusion. And they achieved this with 88% of their patients. However, after treatment with the Bluebird Bio therapeutic, the stem cell that was edited and then given back to the patients, their own stem cells, they had 32 people in this clinical trial. 2 died from leukemia. And they are having trouble saying whether or not it was due to this therapeutic or due to something else. However, the FDA has given them a black box warning and have required that patients are followed up with every 6 months to make sure that they are not developing cancer or leukemia. There were a total of 44 patients in the Vertex trial. None of them developed leukemia. At this point, many of the patients in these clinical trials have been disease-free for years. All of them that have been interviewed have been quoted as saying this has changed their entire lives. And that's well and good, but these therapies come with a hefty price tag. Sit down if you're not sitting down already. The Vertex therapy, $2.2 million. And we're kind of in a new realm of curative therapeutic for chronic disease. And so the pricing here, and it's the first one for sickle cell. So the pricing here is being justified by the pharma companies by saying there's an expected lifetime savings of $4 to $6 million for hospital care, insurance, etc. I still think that is going to limit, if not completely remove, access for most people. Insurance companies really don't like doing massive payouts, but we shall see. It's really terrible to design and build an entire curative drug and not be able to get it to anyone. So I'm looking forward to those costs coming down. Something else to note about these therapies is that patients do have to undergo radiation, and this can be sterilizing. Vertex has said they will offer reproductive support alongside of their therapeutic approach, and it is not offered for anyone under 12 years old. And remember, with sickle cell disease, strokes can start as early as around 6 years old. So this is a massive advance in health and medicine, but it's not perfect. It's really not perfect. And these patients do need to be followed up. We need to see what happens. I'm really curious if we are going to figure out what happened with those 2 patients that developed leukemia, and we will go from there. But there are several people now living a very happy, healthy life as a result of these therapeutics. Also, access. That is a very, very expensive drug. Neither company thinks they're going to have problems with getting insurance companies to pay for it. We will see. We will see how that goes. And I am, again, looking forward to those prices coming down. What's new? What's next for gene therapy and development? Let's see. So there are dozens of active and or recruiting clinical trials that use CRISPR, anything from removing drug-resistant genes from enterobacteria, and a phase 1/2 that's looking at transplantation of insulin-producing cells in type 1 diabetes. So all of these things are kind of new and neat advances. I think there's a lot that can be done. Um, my final word on this is that CRISPR technology really does open up a huge life-changing approach to curative therapeutics, things where you don't have to be tied to injections and pills or fearing, in the case of sickle cell disease, an attack that lasts for a week and really just knocks you out and reduces your lifespan, and you're living a very painful existence. So that is really exciting. But I also want us to make sure that we're approaching things ethically. And I used to be part of a gene therapy club, if you will, when I was a postdoc at one of the universities I was at. And we would talk about emerging stem cell therapeutics. This was based on gene editing, and this was well before the Nobel Prize for this was awarded. And one day, one of the professors that would sit with us asked, what do you guys think the most important element of gene therapy was? And a bunch of us decided that it was an off switch, a way to make sure that if anything does go wrong, you can remove the offending cell type. And so I'm looking forward to seeing if people are developing that as well. So thank you for joining me today on From Bench to Bedside. I hope you enjoyed learning about the history of sickle cell disease, learning about where it came from, how the sickle cell trait is protective from malaria, and that's why it was genetically selected for in 4 different events at least, and how we are approaching this disease that really hasn't had a lot of good treatments in the past with a potentially curative therapy. All right, I'll see you guys later. Take care.